Is everyone? Please let this be a normal field trip. With a friend? No way!
Cruising on down Main Street, you're relaxed and feeling good. Next thing that you know, you see it. What's in the neighborhood? Surfing on a sound wave, swinging through the stars. Take a left at Jimmy Jackson. Take your second right back on to the magic scuba. Alligator nostril. Climb on the magic scuba.
Bro, this is so stressful. When does science get so complicated?
Tell me about it.
Like, what is the Genome Project anyway?
Well, the Human Genome Project was a massive scientific effort to identify all the genes and nucleotide sequences in the human body.
That was a rhetorical question.
Oh.
Like, why would Miss Rizzo even give us this topic?
I don't know. I find it kind of interesting.
Maybe so, if we were there when it actually happened, but like, researching is kind of boring.
Maybe you can be there when it happened.
What do you mean?
I mean, I have an idea. Come with me.
What are we doing here?
We're going back in time.
Are you crazy? You're gonna hijack Miss Rizzo's bus?
It's in the name of science. I'm sure she won't mind.
What do you mean?
Come on. Blink.
Whoa! Where are we?
The 1990s, I
Wait.
think.
When the Genome Project started?
No, geneticists already started research years before it was actually official. In 1987, the Department of Energy's Biological and Environmental Research Advisory Committee recommended the implementation of a large-scale project that would help map out the entire human genome. But the real project didn't begin until...
1990?
Yeah!
Wait, no, the year is 1990.
Wait, what?
This is
Whoa.
sick.
But how are we supposed to get out of here?
You mean you didn't think of a way back?
Yeah, I kind of didn't think of that.
What do you mean? I'd only stuck here for 15 years?
I mean, it was only really 13.
13 is still a lot.
But I mean, it was one of the largest single scientific projects funded by the United States government.
Just because they got $2 million for 15 years doesn't mean we're getting that money.
Haha, very funny. All I'm trying to say is we might as well get some info while we're here.
Where would we get the info?
I don't know. I'm sure this place is buzzing with research centers.
Well, from my research, the National Research Council recommended the National Institute of Health take over the Human Gene Sequencing Program. When the project first began in October 1st, 1990, James Watson was the first director of the project.
You mean the co-founder of the double helix?
Yeah, that guy. But he didn't last very long. In 1992, Francis Collins took over the project.
So, wait, what does this have to do with research centers?
Well, from here, things only got better. The Human Genome Project became an international project that involved at least 18 countries.
Whoa.
And although 3 national genome centers were opened between 1998 and 1999, the actual gene sequencing was carried out in many different research centers in USA, Japan, England, France, Germany, and China.
Are you saying we're in China?
No, what I'm trying to say is if we go back to one of these laboratories, we can probably figure out how they map out all the genes in the body.
Right, that was kind of what I was trying to get at when I mentioned the research centers. But yeah, good idea. How about we go to the Lawrence Berkeley National Laboratory down the street?
Okay, let's go.
Whoa, what do you think they're doing in there?
Probably mapping out the human genome.
I know that, dummy, but I mean how?
Well, I read that in these research centers, the research teams would work towards sequencing a specific segment of the chromosome and then synthesizing the results. Part of the project included mapping and sequencing the genome of other organisms to work towards refining genetic technologies.
Whoa, that's actually really cool. I wish we could go talk to them.
Why can't we? I'm getting bored of watching them work anyway. Let's go.
No! Haven't you watched any time travel movies? You're not supposed to interact with people from the past. It messes up the entire time and space continuum.
I guess you're right.
But speaking of time, I should probably head back to the bus and see if there's like a reset button or something.
Okay, you do that.
I'll call you when it's done. And remember, don't talk to anyone.
Yeah.
I'm serious.
Yeah, whatever. I'm gonna go into the laboratory. I don't care what she says. Hey.
Hey.
What are you guys doing here?
Well, we're trying to sequence out the entire human genome.
What does that mean?
We are going to identify and map out all the 30,000 genes and 3 billion base pairs of nucleotides that make up a human being.
Wow! How do you correctly determine the correct order of bases?
In order to accurately determine the order of nucleotides, scientists sequence a chromosome multiple times. And in order to sequence each chromosome multiple times, we need multiple copies of one gene. These copies can be produced using bacterial plasmids or through polymerase chain reactions.
Oh my god, I remember researching this! When you isolate a bacterial plasmid and a section of DNA, you can use specific restriction enzymes to cut both the plasmid and the strand of DNA. These restriction enzymes produce sticky ends where the plasmid and DNA can be joined together to form recombinant DNA. The recombinant DNA is then inserted back into the bacterial cell, and the bacterial can multiply multiple copies of the same gene. Am I right?
Yes, absolutely. And although this method has been used, PCR is used more often as it provides multiple copies of DNA at a much faster rate. Do you know how PCR works?
Now that you mention it, I think I do. In PCR, a sample of DNA is heated so the DNA separates into two pieces. Then an enzyme called Taq polymerase synthesizes the two new strands of DNA using the original strands as templates. These strands are then used to create two new copies, and so on. The cycle of denaturation and synthesizing new DNA is repeated as many as 30 or 40 times, leading to more than 1 billion exact copies of the original DNA segment.
Exactly. From here, there are multiple sequencing techniques that can be applied. Some of these techniques include the Maxim-Gilbert technique, shotgun sequencing, and Sanger sequencing.
Can you tell me a little bit about them?
Sure. In the Maxim-Gilbert technique, both strands of the double-stranded DNA are labeled using radioactive phosphorus. The DNA is heated, causing both strands to separate. These strands are then cut up into a number of shorter pieces using specific restriction enzymes. The differently sized fragments of DNA can then be separated using gel electrophoresis, and the resulting patterns determine the sequence of each DNA strand. In shotgun sequencing, a variety of restriction enzymes are used to cut the DNA in different areas, producing hundreds or thousands of random bits. Each small stretch of DNA is automatically sequenced, and then powerful computers piece back together the information to generate the entire DNA genome sequence.
What about the Sanger sequencing technique?
That one's a little complicated. It involves combining a DNA sample with a primer, DNA polymerase, DNA nucleotides, and small amounts of the 4-dyed labeled chain-terminating dideoxynucleotides in a tube.
What are dideoxynucleotides?
Unlike deoxynucleotides, dideoxynucleotides lack a hydroxyl group on the third carbon on the sugar ring. The absence of the hydroxyl group causes the chain to end with a dideoxynucleotide, which is marked by a specific color depending on which base it carries. The mixture is then heated to separate the DNA strands so that the primer can bind to each single-stranded template. The temperature is risen once again to allow the DNA polymerase to synthesize the new DNA until it adds a dideoxynucleotide instead of a normal one. At this point, the strand will end with a dideoxynucleotide because no more nucleotides can be added. This process is repeated for a number of cycles. In the end, the tube contains fragments of different lengths, each ending at the position of the dideoxynucleotide on the original DNA strand. After the reaction is complete, the DNA fragments are run through a tube containing a gel matrix in a process called capillary gel electrophoresis.
Wait, you talk about gel electrophoresis a lot. Isn't that when you place a substance into a gel matrix and then run an electric charge through it? This separates the substances based on size? Am I right?
Yes, you are. Only this time, when each fragment crosses the line at the end of the tube, it is illuminated by a laser which detects the color of the dye. The detector produces a chromatogram which shows the fluorescent intensity in a series of peaks. The DNA sequence is then read from these peaks.
Oh, that makes sense now. You can make multiple copies of a target gene using bacterial plasmids and PCR and then determine its sequence in a variety of ways with the help of restriction enzyme and gel electrophoresis.
Yeah, if it were only as easy as it sounds.
True, but I'm kind of curious as to why you guys are trying to sequence the human genome though. Like, what's the point?
Well, the completion of this project will help scientists understand the organization, location, and expression of genes as well as how they are duplicated, inherited, and mutated. Through the Human Genome Project, we hope to locate all the normally functioning genes as well as those that cause specific illnesses. Knowing the nucleotide sequences can help determine the physical basis of hereditary illnesses as well as those for normal growth and development. The human genome will help health professionals around the world identify root causes, diagnosis, and treatment of a wide range of genetic illnesses. Not to mention, the completion of the Human Genome Project would improve our quality of life in general, as it would expand our understanding of other organisms such as bacteria that could possibly benefit our economy, lead to cures, and reveal secrets about human evolution.
As great as all that sounds, I'm still a little skeptical about how mapping out the human genome will all help with all these things.
Well, with the completion of the Genome Project, researchers will be able to find genes associated with certain diseases, like breast cancer, thus making it easier for doctors to diagnose these conditions at a much earlier point.
Wow, that's great. But how would it help find new uses for microbes such as bacteria?
The knowledge gained from the Human Genome Project can be used to understand the genomes of bacteria, which can then allow us to understand how harmful bacteria work against the body and how to combat them. Understanding the makeup of bacteria can also help with useful applications such as energy production, environmental cleanup, toxic waste reduction, and new biotechnologies that involve bacteria.
What does the Genome Project have to do with evolution though?
The Human Genome Project will help scientists better understand evolution by comparing genetic makeup of humans to other living things. This can help us understand more about our species in comparison to others.
If we are able to determine our genetic makeup, then can scientists use it for forensic science when it comes to DNA profiling?
Exactly! Techniques developed during the Human Genome Project have made it possible to create a DNA profile of a person using DNA fingerprinting. You can then use these techniques to identify victims of a crime, match an organ donor with its recipients, and even determine the father of a child.
Wow! That probably would have been really useful in Star Wars.
Yeah, I guess you can take it like that. And finally, having a deeper genetic understanding of plants and animals, as well as humans, allows farmers to develop crops that are disease, insect, and drought resistant. So even though the project might have cost a lot to begin with, it's actually very beneficial to our economy.
Wow, I never realized how many applications there are to the Human Genome Project. But don't you guys think that there are going to be some ethical as well as social issues that will come from this?
Such as what?
Well, some companies would be able to create artificial DNA and get them patented. This basically gives those companies ownership of the DNA and they can decide who gets the gene and who does not. Also, insurance companies would be able to use this technology to look into an individual's genetic information to determine if the person is eligible for health insurance or not. This would cause many complications as individuals that are likely to have genetic diseases would be denied by insurance companies. Also, genetic scientists would be able to detect diseases and genetic defects in a fetus very early on. This would give families the ability to change and modify the genes of the baby even before it's born. Many groups and religions believe that the use of such genetic technologies is unnatural. They would argue that humans should let nature play its role and that humans should stop playing God. Additionally, 99% of the genome is essentially the same. 1% is different. This 1%, however, codes for roughly 3 million bases. I'm not a genius, but that sounds like a lot of bases. Not to mention, sequencing the human genome could lead to the development of threatening biological weapons, genetics discrimination, and human cloning.
I guess you're right, but that's nothing we haven't already thought of. At the beginning of the project, we set aside 5% of the project's budget to fund discussions surrounding the ethical, social, and legal implications of the project, thus making it one of the first big projects to address any ethical concerns.
Oh well, at least something is being done to address these issues. I should probably let you get back to work. By the way, do you know any way I can get back to 2018?
Let me think. Well, there is one way. You would have to travel to each significant year of the Human Genome Project. Once you travel to each significant year, you will be able to go back home.
Okay, thanks. Bye.
Bye.
Hey, actually, I know a way we can get back to 2018.
How?
So we have to go to each significant year of the Genome Project, and then we'll be able to go back home.
What are we waiting for then? Let's go!
Let's go.
In 1992, the very first rough map of the human genome was published using data acquired by linking known genes and their locations.
In 1994, the Human Genome Project announced the completion of its 5-year-old goal of producing the map of the human
Was
genome.
passed called the Genetic Privacy Act, which was created to control how DNA is collected, analyzed, stored, and used.
1995 was a big year as the physical maps of many chromosomes were complete. Maps of chromosomes 16 and 19 were published, followed by the maps of 3, 11, 12, and 22. At this time, the genomes of many other organisms were also being mapped. What's a physical map?
Well, when sequencing the genes, they made two types of maps. Before sequencing, a physical map was used to measure the distance between two genes in terms of nucleotides, and a genetic linkage map was used to measure the distance between two genes in terms of how frequently the genes are inherited together.
Oh, now it makes sense.
Yeah, now shush, I'm trying to listen to history.
The scientific community, concerns over discrimination based on genetic information started to arise. This caused the Healthcare Portability and Accountability Act to be created, which prevented healthcare insurance companies to use genetic information in certain cases to determine eligibility. In a 1996 summit in Bermuda, leaders of the Human Genome Project agreed that all data found around the world would be put online within 24 hours of discovery so scientists across the world could access the information. This was known as Bermuda Principles. These rules were made to ensure that the Human Genome Project was not a race, rather a collaborative effort of scientists all around the world. In January of 1997, the NIH declared that the National Human Genome Institute was a recognized collaborative institute. Following this, the physical maps of chromosome X and Y7 were announced.
In 1998, a gene map was released that allowed scientists to use the map location of approximately 30,000 genes for genetic studies. 1998 was also a significant year as J. Craig Venter, the founder of Celeria Genomics, declared that his company would sequence the human genome on its own, using the largest civilian supercomputer capable of producing the needed sequences. To many, J. Craig Venter's progress came across as a race between the public and private sectors. However, when Venter met Francis Collins, they agreed that cooperation would achieve more than competition.
A scientific milestone was achieved in December 1999 when an international team announced that they had completed the entire code of chromosome 22. Chromosome 22 was relatively small in comparison to others and is linked to major diseases and has a sequence that consists of over 23 million letters in length, making it the largest continuous stretch of DNA ever deciphered and assembled. To see a human chromosome sequence for the very first time is like seeing an ocean liner emerge from the fog when all you've ever seen before are rowboats. On June 26, 2000, Venter and Collins both announced the completion of a draft sequence, a rough copy of the human genome whose sequence was about 90% complete.
In 2001, the complete rough copy of the entire human genome sequence was finally published. Even after it was published, a lot of effort went into verifying its accuracy. Before 2001, many scientists believed that there were about 100,000 genes in the human body. However, the new research found that there were only about 30,000. After this discovery, scientists concluded that the human complexity does not come from the quantity of genes, but rather as a result of the structure of the network of different genes, proteins, groups of proteins, and the dynamics of those parts connecting at different times on various levels. Even though the human genome sequence was completed in 2001, work did not stop. In 2003, a higher quality sequence of approximately 95.8% of the human genome was published. This version was very accurate, with an error rate of only 1 base per 10,000 bases. Whoa, I can't believe the project is over!
I know, but project's not over for us. 2018, here we come!
So, what do you think?
I have to admit, the Genome Project is pretty amazing. It kind of has a lot of ethical issues, but there are also a lot of benefits.
You're so right. The project was very well thought out, and it had a rich history full of great scientific technologies, and above all, cooperation.